welcome to the Duke CABC Center on Addiction Behavior Change seminar series. I'm Ed Levin, Director of CABC, and we are just coming off our spring symposium, which went very, very well. And before I introduce today's speaker, I just want to look ahead a little bit. In a couple of weeks, we're going to have Byron Jones from the University of Tennessee come in and talk about his work in ethanol and pharmacokinetics and pharmacodynamics of ecstasy. So the interaction between two different drugs of abuse. And then after that, a week after that, on March 23rd, actually a former student of mine, Reggie Kennedy, who's now a faculty member at North Carolina A&T, is going to be talking about his work, again, with ethanol and looking at the cellular substrates of heavy drinking chronic alcohol. And then Emily Rissman from NC State is going to come in April. And then finally, we're going to have a co-sponsorship with the Duke Center on a study of adolescent risk and resilience. Dr. Nisha Godfreyson is going to speak from North Carolina, UNC Chapel Hill. So now I'd like to introduce today's speaker, Dr. Linda Porrino, who is really one of the world leaders in the area of the neurobiology of drug abuse. She earned her undergraduate degree at Bates College in Maine, and here on these notes, it said received her PhD. Nobody receives their PhD. She earned her PhD in psychology from New York University.
Well said.
And so, and then she worked as a postdoctoral fellow at National Institutes of Health, collaborating with Dr. Patricia Goldman-Rekeach. And then she carried on in brain imaging and surgical neurology branch as chief at NIH. And then she has been chair and now is emeritus at Wake Forest University Department of Pharmacology and Physiology. And I'm looking forward to hearing about her perspectives on drug addiction and research. Linda.
Oh, thank you. Okay, so I will try and do this, and hopefully it'll work. Okay. Well, I want to thank you for inviting me. This is really a pleasure to come and meet up with some old friends and see some people that I haven't seen in a long time, albeit not exactly the way I would like to, but it's kind of, I guess Zoom is better than nothing these days. So I want to use this talk to talk about some of the work that I've done over the years and trying to translate the studies that we do preclinically to studies to the human condition. So as I said, some reflections on a career in translational neuroscience. I want to thank my lab over the years for all the work that they've done, because they are some of my pictures of my lab over the years and its various forms and whatever, but these are the people who really do the work. I just sit and write grants so that they can do the work, as we all know. So I want to talk about cocaine for the most part. My lab over the years has really studied the consequences of chronic substance abuse, whether the substances were therapeutic substances or recreational substances. But the largest portion of my work has really been done on cocaine, which despite all the media attention on other drugs like opiates and cannabis, still remains a rather consistent and problem. And there are still a large chunk of people who use cocaine and are still dying from cocaine overdose, particularly now that we're now lacing our cocaine with fentanyl. This has become an increasing problem. So continuing, but you know, despite the fact we've studied it for years, and I certainly have studied it for years, we still have no good way of treating it, and we haven't made much of a dent in the numbers of people who continue to use it and experiment and become users over the recently as they grow as we move along in years. So we have no way to really reduce, excuse me, the harm of cocaine use disorder. So over the course of my career, over the years, I've asked a bunch of questions about cocaine, and I'll try and address each of these today in my talk. And I think the first and most important question was always whether the deficits that we see in drug users are really the result of cocaine exposure, or whether these deficits predispose them to use drugs like cocaine. So that's one question. And then another important question, if they are the result of drug exposure, where and when do these changes develop? And most important question that I think we need to ask, and we need to ask more and really study more clearly these days, is the question of recovery. And I'll try and address some of that as I go along. But given my early training as an anatomist, which Ed alluded to earlier in his introduction, most of my work has really focused on the where things occur, and rather than the how and exactly what changes occur. But we were very interested in always looking at where. So I've used over the years a whole slew of imaging methods, from in vitro methods and animal models where we did metabolic mapping with the 2-deoxyglucose method, where we measure glucose utilization as a reflection of functional brain activity. And the advantages of these techniques are obviously that we can look exactly where something occurs. And we done huge amounts of receptor autoradiography and all kinds of, again, looking at the where. But in vivo techniques, we've done mapping of blood flow with functional magnetic resonance imaging, fMRI, or glucose utilization now with PET. And the obvious advantages of those are that they're non-invasive. We can study humans, and we can also do longitudinal studies. I'm not going to talk about much of our longitudinal studies today, but it's possible to look repeatedly in the same individual over time when you're doing these kinds of in vivo methods. But to answer the questions that I wanted to ask about the deficits that exhibited by cocaine users and where and when these occur, these are very difficult questions to answer in humans. Obviously, humans tend to not be particularly selective in the drugs that they use. They tend to use all of them, and it makes it very difficult to study one. They also have all kinds of other issues that confound any studies that we do, and it's very difficult to study, obviously, to study humans in the earliest phases of their drug use. They don't generally come to us until they're long gone. So to answer these questions, we've used an animal model. And in our case, we've chosen to use self-administration in non-human primates. We use primates because of their neuroanatomy, their similar neurochemistry. And I take that one as a very important piece of why we use primates. Humans have somewhat very different receptor distributions in certain cases, and that are important, particularly in the opiate field. I mean, humans and primates are very different than rodents. They also have very similar cognitive skills. We can look at systematic evaluation of variables. And after my experiences in human studies, the very important part about working with monkeys is they always showed up for their studies. They never failed us, which was my important thing. So I'm going to talk about a series of studies that I did with my colleague and collaborator, Mike Nader, who has I've worked with for, believe it or not, over 25 years. And Mike and I have done a large number of studies, but I'll talk about this series of studies in which we looked at cocaine self-administration at 3 different time points in the history of cocaine use, and trying to model different phases of the addiction cycle, so to speak. And so I'll start by talking about the fact that these are monkeys that self-administer pretty regularly. They self-administer a relatively high dose of cocaine for a monkey, and have rather extensive histories by the end of the 500 sessions, but I'll get to that. But we study them, so we allow the animals to self-administer for their daily session, and then at the end of that session, at the end, when there are in the darkened chamber, when there are no more cues, we apply the 2DG method, and we sacrifice the animals at the end of that. So I'm going to go over pretty quickly what goes on after 5 sessions. And what we see, and I'm going to focus on the striatum. Oops, there. Okay, so the cocaine's effects during the initial or recreational phases, when they're just getting, when they've used the drug a few times, in this case 5, but here's the example of some pictures of the brain sections through 3 levels of the striatum: the most rostral portions, the central portions where the caudate and putamen are very much differentiated in the monkey, and this is the ventral striatal areas down here, and then this is post-commissural, where the striatum is largely receives all of its inputs from more sensory motor areas, and is really more sensory motor in function. More rostrally, it's from prefrontal cortex and orbitofrontal cortex, so it's involved in cognitive function and valuation, while here at the mid-range, this is areas, again, prefrontal cortex, a lot of inputs from particularly orbital and medial cortex in the ventral striatum, where you see that cocaine is having these blue blobs indicate that it's decreasing decreasing brain functional activity in these most medial and ventral portions of the caudate, as well as in the ventral striatum, the extent of the ventral striatum. But it's absolutely restricted to a very small portion of the striatum. But then, whoops, and then here's a schematic that you'll see repeatedly. So here's the blue again indicates decreases in functional activity, so decreased brain activation in prefrontal areas and in some of the caudate, and again in the striatum, a bit in the thalamus, and it's in the medial dorsal portions of the thalamus, as well as in the hippocampus. So we're seeing that it's relatively restricted. It's occurring at very specific locations. These are mostly areas that are involved in the processing of value and emotion, motivation, and some cognitive places as well. So what happens after longer periods of time? In this case, the animals self-administer for over 3 to 4 months, and now we are sacrificing them. So we're looking at the effects of 100 sessions of cocaine, so a whole series of cocaine exposures and experiences, mixed in with the acute effects, combined with the acute effects of cocaine during that final session. So what do we see now in the striatum? So comparing it to the initial phases, you can see that the blue blobs have grown dramatically. They're much bigger. much wider expanse of the striatum is involved, particularly the entire caudate, moving into the putamen, more rostrally. So it's moving dorsally and rostrally as in the effects. So cocaine is having a much broader effect now and much wider expanses of the striatum. And that's true for the rest of the brain as well. The larger portions of cortex are now involved and have decreases in functional activity, as well as large portions of the striatum, larger portions of the thalamus, hippocampus, and now even the ventral midbrain striatum, the dopaminergic regions that project to the striatum and basal ganglia as well are starting to be involved. And we're starting to see decreases in larger numbers of areas. So the effects of cocaine are spreading. But what happens after 500 sessions? Now we're talking about really prolonged self-administration, and we're starting to live, this is over 500 sessions over a 2 to 2.5 year period when they've had these experiences. And this is now the amounts of cocaine, total intake are approaching some of the levels that we see in some of our users that we see in the lab in terms of cocaine history. So now we're really looking at a more extensive experience. And what do we see? We see that it's even more progressive, and that cocaine's effects are spread even more widely throughout the entire striatum. There is no portions of the striatum that are really left untouched. And that's true for a large portion of the brain as well. Large portions, expanses of cortex, ventral midbrain, thalamus, amygdala, hippocampus. There is cocaine's having decreases brain activation throughout the entire brain. The effects of cocaine, although very restricted early in its effects, really influence greater and greater portions of the striatum and the whole brain as use becomes more prolonged. The expansion actually follows a very clear anatomical trajectory. And although I didn't show you this, it's true for dopamine receptors, dopamine transporters, actually opiate receptors as well. There's a progressive dysregulation of all of these receptors and transporters with that follows a very similar anatomical trajectory. So one of the important implications of this kind of expansion is really that in terms of thinking about the preclinical models that we use in many of our studies and use to develop potential therapeutics for cocaine abuse is really, this has some important implications in that most of the studies that we do really focus on the very early phases of cocaine self-administration, so that people, we tend to make decisions about whether drugs might be useful in the clinic based on the very earliest phases of cocaine self-administration. And cocaine has much broader and much bigger effects than that, especially when you're talking about users that have been using for 10, 12, 15. Some of our users have used for upwards of 30 years. I think that trying to get recovery and trying to find drugs that are therapeutically beneficial is not going to be very effective if we're only looking at models and trying to pick drugs based on models that I don't think really mimic the real cocaine user. But more importantly, we, in terms of our questions about cocaine, I mean, we see this in animal models, but is this true in humans? I mean, we really don't know the answer to that. And how can we address this question in cocaine users that have been using cocaine for years? And supposedly, if we're right, then they should have had this expansion. So our hypothesis was that if the effects have expanded, then regions of the striatum beyond the motivational and cognitive portion should be impacted. So we should see sensory motor effects, do we? And that's the question that we asked with my colleague, Colleen Hanlon, who worked in my lab. It's actually a Duke grad from your neuroscience program. And we did studies in a bunch of users and controls. Cocaine users that you can see have been using, I think there are about 30 all told in this study, and about 30 controls. And they've been using for very long periods of time, 10 years or so on average. And they used fairly regularly, 3 or 4 times a week. And so we did a study using fMRI in which we asked the subjects to perform a very simple motor task. This is something that is is used for demonstrating MRI effects all the time, how MRI works all the time, tapping a finger, and this is what it looked like. So we showed them a screen with people, you can see the tapping, and we did a predictable task in which they did the same thing over and over again, unpredictable, where they had to follow the sequence that they saw on the screen. So how did our cocaine users do? So here, the controls are in yellow, the users are in blue. You can see in terms of latency, they're a lot slower than our healthy controls, and they make a whole lot more errors, both in the predictable task, relatively straightforward and easy, just tapping fingers over and over again, but also in the unpredictable task where they made huge numbers of errors. So what did their brains look like when we were doing this? And you can see, I'll only show you this from the unpredictable task because I think it's kind of the most telling. But you can see here in the normal controls that, and in the healthy controls, that the finger tapping elicits the very like this is classic ipsilateral, contralateral. They're all tapping with their right hand, and this is activation in the left in cortex and also in the left striatum, which I'm showing you here. When you look at the cocaine users, however, you can first of all see that they're not, the activation is much less. They didn't quite get up to the activation in the controls, but also that it's much more widespread. And I think that's a particularly interesting thing that we fondly called in the lab sloppy brain, but it indicates that wider expanses of the brain resources need to be recruited in order to perform the task. So instead of being able to use a very punctate region of the striatum and the cortex, these subjects have recruited a wider expansive brain, in fact, using the contralateral hemisphere as well. So not only do we see this wider effect within the striatum and cortex, but we also see the loss of laterality as a result of the cocaine use. So in our cocaine users, while they're trying to perform this task. So sloppy brain lives. So our chronic cocaine users clearly show impairments in sensory motor function, and they're accompanied by disrupted activation patterns in the striatum as well as in the cortex. And these impairments are correlated with the duration of cocaine history. Those subjects that had used the longest made the most mistakes, and they also showed the most disrupted patterns of activation in the fMRI scans. But so we talk about a phrase that I actually got from Nora when she saw some of my results. She called it territorial imperative of cocaine, and that we see in our preclinical models also occurs in human users. But so we've answered the questions in some respects of that. Is this due to cocaine? Yes. We've looked at the where things cocaine has its consequences, and so we've answered our first two questions to some extent, anyway. But the real question that I want to focus on is really the question of is there recovery, and are these effects that we see, the changes in the brain, permanent? Do some of these effects reverse? If so, what is there a trajectory to that reversal? Does it follow the same anatomical pattern of development? Does it revert back? Do some effects remain? And in fact, what is recovery? Relapse rates are extremely high, but some people do manage to quit. And what do their brains look like? And should we be looking at their brains to get a hint about how what we should aspire to in terms of treatment? How do they get better? And how do they manage to stay? What resources do they have and use to reverse the effects that and be able to remain abstinent and recover? So I'll first talk about the animal work that we did with Mike Nader. And in this case, we were looking at that same type of experiment where the animals self-administer for long periods of time, and then we do our 2DG procedure and sacrifice the animals. And in this case, the animals self-administered for 100 sessions, this mid-chronic effects, where by the time 100 sessions has gone by, the vast majority of effects have occurred in terms of the involvement of the striatum and the cortex and other areas in the brain. And then, but instead of sacrificing them after cocaine use, they had been withdrawn or ceased cocaine use for at least a month. And we then looked at them at this point. And in these studies, all the animals are in chambers in which they've never received cocaine. So there are no cues around. There's none of the rest of that kind of environment. These are chambers in which they've actually only spent time looking at videos on Disney movies for all their time. And then they go on and so they're in a darkened chamber, and we're trying to approximate what we call basal brain function. And so this is what we think the animals then bring to their environment. So this is their basal state. So let's look at what happens. And this, so these are schematics from, actually taken from a paper that we just published, outlining these effects. And so you can see the blue indicates decreases in functional brain activity. And you can see wide expanses of the cortex, the striatum, the hippocampus, the thalamus, the ventral midbrain are all very much impacted after 30 days of abstinence. These are still remaining, still functionally decreased as compared to controls, food reinforced controls who follow the exact same, had the exact same history, except they don't have any cocaine. And so you can see that these guys, that the effects of cocaine are very persistent and do not tend to decrease with time. In fact, one of the things that we looked at was we looked at a group of animals after only 1 day of abstinence. And you can see that there are rather substantial decreases there as well. But that if anything, the effects after 30 days have grown even more intense, and brain is even more disrupted after 30 days than even after 1 day. So that this is not exactly a good sign in terms of the permanent. I mean, the fact looks that many of these effects really are very much permanent and continue to grow over time rather than reverse or decrease as we would hope. Oops, I'm going in the wrong direction. So here, again, the same schematic, and these are the effects that we see. So effects throughout cortex, all areas of cortex that process all varieties of information, including motor information, cognitive information, attention. Parietal cortex is very much affected. Large effects in thalamus, particularly in mediodorsal thalamus and its connections to prefrontal cortex and cingulate. Hippocampus, meaning that there's likely impacts on learning and memory, as well as throughout the vast expanse of the striatum. So we're seeing that cocaine's effects are not going away, as we would hope after time. So as I've shown you, the impaired functional activity is still present despite 30 days of abstinence. And again, there are no cues present. So this is not the result of the fact that they expect cocaine in any way. They've been in this environment many, many times. And in fact, during the time that they're abstinent, so that, you know, we can say that there's very, very little evidence for recovery at this 1 month time period. If anything, it appears that the functional disruptions grow after time. So if we're going to expect recovery and some reversals, 1 month is clearly not enough. It just doesn't seem to be what we hoped it would be. But so, you know, in thinking about recovery, we really, you know, a very common treatment model in medicine is you identify an abnormality and then you work to fix it and to normalize it and to bring it back to what it was. But we know that the brain is likely much more complicated than that. And recovery or remission might require some adaptive compensations that don't return the brain to its normal state prior to the insults. So are we looking for the wrong thing? Are we looking for just are we looking for the effects to just reverse? Maybe that's the wrong approach. And we should be thinking about this in a very different way. And perhaps one of the things that we should think about is whether, like after a stroke or some kind of traumatic brain disorder, there's compensation, and that you know, the brain learns to overcome the deficits that it has. So that's the question that we really want to ask. And is that the case? And can we identify potentially states where this might happen? And so for these studies, this is a series of studies that we did with actual cocaine users that I did again with Colleen Hanlon. And in this case, we're looking at 38 abstaining. In this case, they're all treatment-seeking cocaine users. They're all abstinent at the time of study, and comparing their brains to control brains and the brains of controls. And you can see that our users are relatively standard. They've used for average of 11 years with a wide range, but they actually all have used for at least 4 years in this case. So all the subjects are heavy cocaine users. They use 4 or 5 times a week. That's a pretty hefty dose. Otherwise, and we divided the abstainers into 3 groups. And we have a group of abstainers who abstained for less than a month when we tested them. And we tested them so at least a week, but less than a month. And then we had a group of users that we recruited from local treatment programs. So they're all motivated users. The abstainers were 3 to 5 months, so we called those our midterms. And then we have some long-term users that have been abstinent for as much as for greater than 10 months and as much as 2 years in that group. And we looked at their brains using glucose utilization, in this case, FDG with positron emission tomography. And we did this because fMRI is very good at showing differences, but it's not very good at showing the basal state. And we were very interested in whether how brain function was definitively impacted in this short term, in these abstainers over time. So let's take a look at the short-term abstainers, which look very similar to our monkeys. There are wide expanses of cortex in which brain function is decreased, indicated by the blue blotches. So lots of effects in the cingulate, medial cortex, superior frontal, inferior frontal, gyruses in these areas of the brain. But what happens after midterm? Now we're talking about 3 to 5 months of being abstinent. And you can see that the blue is starting to reverse. So the hope is there that we are seeing some reversals of some of the effects that we see, and that it's really a matter of time rather than that these effects continue to grow. So we're starting to see some of the reversal that we had hoped to see in the animals. And then if we look at the more caudal portions of the brain, we can see extensive effects in cortex and thalamus, and even there are some effects in cerebellum, but also in the brainstem. So that the short term, the effects on glucose utilization or brain function are rather profound, extending throughout the cortex and also subcortically. But again, when we look at the midterm and these after 3 to 5 months of abstaining, you can see that many of these effects have gone away, although there are still effects in hippocampus and thalamus. Subcortical effects seem to stay around a lot longer than the effects in cortex. But there seemed to be clear reversal, which is the good news. But I've shown you 2 sets of effects. Now let me show you what happens in our long-term users, long-term abstainers. So these are people that we would consider in recovery. So instead of seeing blue on this thing, you see blotches of orange and red and yellows, indicating increased activation. Again, we're comparing these all to healthy controls. So it looks like these subjects have recruited, have done super activation and are using their brains in a much higher rate and intensity than the controls. And that's present throughout the areas of the cortex that you can see. You see a lot of the parallels. So those areas in which there were decreases now look like there are extensive increases in brain function. And so let's look at the posterior portions of the brain, and you can see exactly the same thing in these areas of the brain. So you can see these throughout the cortex. We see splotches of this orange indicating increased brain function as compared to the controls, although you might see still some blue patches in the thalamus that seem to persist. In fact, I had one user to whom I showed these results and said, told her what a great job she was doing. And she told me, but that indicated that she's always going to have a hole in her head, and that she would always have that she had to overcome throughout her recovery. But, again, it looks like that long-term abstention leads to increases in brain activity, which, and here's another view of this, comparing the short-term users, excuse me, and the long-term users. Again, the parallels between the places in which there were decreased activity and the places where there now increased activity is very striking, to say the least. So parietal, cuneus, throughout the brain, medial temporal gyrus, we see these large increases in brain activity. So potentially, recovery might be one that increased activity in cortex might be an important characteristic of recovery. And if we are to see recovery, that we may need to have increased function in cortex. So I think we've learned a lot from these human studies, and that we can see that abstinence can reverse some of the neuroadaptations that accompany drug exposure. And that's very good news. But of course, there are important implications for that. Most of the treatment programs that we use in our medical model tend to focus on short-term abstinence, and we need to, it is likely that we need to support people for much longer if we're to encourage real abstinence. And finally, some of the effects appear more permanent. There are effects in the brain that don't seem to be recovering as quickly, particularly in areas of the thalamus and hippocampus. These do not appear to be going away as quickly. And finally, recovery may require active compensation in cortical areas to overcome longer lasting deficits in subcortical regions. That's our interpretation. So I think the implication of this is that we're really looking for ways to enhance brain activity in cortical regions may hold the key to decreasing relapse rates. Can we find ways to increase cortical activity? Can we find drugs particularly that may do this, or potentially the new use of neuromodulation techniques? I think TMS, may get my abbreviations mixed up, but TMS is one transcranial stimulation may hold a lot of promise, that if we can find ways to enhance brain activity, that we may be able to finally decrease some relapse rates. So many years of work over the years, and I think we're starting to begin to understand what recovery means and whether recovery is possible, and trying to get a picture of what it looks like. There's a whole lot more work that's being done in this area these days, and I think it really holds the key to the future in being able to find ways to finally start to decrease the relapse rates that we're seeing with cocaine use disorder. And so with that, I will say many thanks to many of the people that have worked at my lab, and as well as all the funding that I've received over the years from all the agencies that have deemed my work worthy of some support. And I'd be glad to, I hope I've left enough time for questions, and I'm glad to answer any of them that you may have.
Well, thanks. That was marvelous. And yeah, we do have time, and you gave a wonderful overview and some very cogent points. Yeah, we have about 15 minutes, which is great. I have an initial question. In terms of enhancing recovery, the drug treatments to date that have been developed for addiction have been really focused on just kind of preventing relapse on a kind of a primary battle in terms of the, decreasing craving or decreasing using. But you alluded to, other sorts of treatments, they might be magnetic, they might be drug, they might be some behavioral practice or something. So do you have any leads in terms of what might help rehabilitate this lost function, either through restoring that function or having other brain circuits kind of take the place?
So, I talked about TMS. That's something that I think is really very, very promising, and has shown a lot of, not just for cocaine, but it's now approved for use in with nicotine, of course, and so smoking cessation. It's a tool in smoking cessation. I think that we need to explore this a whole lot more, and think about various regions, areas of the brain in which we can do this. I mean, so far, most of the work that's been done focuses on prefrontal cortex, but it may be possible and may be necessary that we need to apply some of the TMS more broadly, and in fact, increase activity in other areas of the cortex as well that might have consequence. But I think, just focusing on drugs that tend to supposedly reduce the craving piece is not enough. And if we're going to get real recovery, we have to do a whole lot more. And so, yes, treatment programs need to be a whole lot longer than they are. And we need to do a lot of cognitive behavioral therapy and training to retrain the brain to do what it's supposed to do.
Okay, well, thanks. Cindy, you have a question?
Cindy. Hi, Cindy.
Hi, I'm so excited to see you. This is so
I know.
And I have two questions. The second has a comment associated. First of all, did any chance you did the finger tapping? Do we know if they recover functionally as their brain activity changes?
So we didn't do finger tapping in those guys, but we did a lot of cognitive tests and cognitive testing, and there is a fair amount of recovery. And the other thing that's interesting is that we did a sort of very crude measures of depression and anxiety in these guys, and they had far less than any of the other our other subjects. So we do see recovery of function, definitely. Although we never did the we didn't do the finger tapping.
No, but it's so wonderfully simple. I always try to think of really simple-minded things to do. With these people, my other question, because this is these are related. I always get frustrated by oxygen utilization because I always want to know what changed, because it's just like something miraculous changed and what changed. And were these people getting therapy at the time? Were they getting cognitive behavioral therapy? Do you know? Or were they just in passive withdrawal and this is the brain recovering on its own?
So all of these people were in treatment programs, and some of them were in residential treatment programs, and our long-term abstainers were in a residential treatment program. And so they were getting therapy, so generally group therapy. Also, in the residential treatment program, they had to earn their way, so they had to work. So there was
I think I know what
an
you're
active
talking,
work
what you're
program.
talking about.
An active work program in this case. And the shorter term abstainers, the all came from either inpatient or outpatient treatment programs. So it's a mix, and all of them involved some kinds of group therapy and frequently work related. They were all tested frequently also, so we knew.
Oh, sure.
Really
Well, you know, it's I hate using. I've always hated the idea of using imaging as a biomarker, but it would be a way to look at what programs were being effective, especially if you could pair it with really simple cognitive testing, which you can. So
yeah.
What circuitry, neurochemically, whatever, do you think was your best guess about what's changing? And I know you have to speculate, but I would love your wisdom.
So, again, I mean, I think the data sort of tell you that it's the secrets in the cortex. I mean, do you want me to tell you which portions of the cortex I think are the most important?
No, I want to know what
that answer. But I do think one area of cortex that's frequently ignored, that I think is probably much more important in all this than we've given it proper attention to, because rats don't usually have too much. And we don't generally know that much about it, is the parietal cortex, and involved in attention. And that I think is a very important place that I'm trying to convince Colleen that she should try TMS in the parietal cortex to see if it had any effects on attention, because I think attention is something that needs to be, needs to improve in these subjects, because they all have very poor attention.
I'm very excited. I have to lecture to medical students, and I'm always telling them, when the person walks in the door, their brain is so adapted, because it's all the way through a whole series of changes, and it's wonderful to be able to also add some hints of the things that will change back, besides D2 receptors, which we know do recover.
Well, the hope is one of the limitations of our study is that it is a cross-sectional study, so that we're looking at subjects at various, so it's separate groups of subjects, and it may be that only those subjects that, only those participants that have, intact brain function are the ones that actually can stay abstinent. But, we did go back and follow some of the subjects that were in the 1 month and 3 to 5 month period. And, we found that some of them had relapsed, but also some of them had remained abstinent. So, we don't have enough data to be able to do a predictive study, but it is pretty, I think it's a limitation that we have to take into account. But again, it's a clue to give us an idea of where we should be looking, rather than and trying to work.
Well, and it's hopeful, because what so many treatment programs tell you is your brain, I always hear the cucumber pickle thing, is your brain has changed and it's never changing back. And I've always thought that was a really biologically untenable and very unhelpful message.
No, I think it's a terrible message, and I don't think it gives people a chance to, that my favorite participant who told me about the hole in her head, she understood that there may be some things that she couldn't fix, but there are also things that she could, and she could adapt. And, people who have a stroke recover. There is substantial recovery after many strokes. I mean, not all of them, obviously, but and there are deficits, but people can manage, and we need to think about it more that way than we do just trying to fix the nucleus accumbens. So.
Really nifty. Thank you.
My own personal.
I'll shut up for a minute. Let somebody else get a word in edgewise.
Thanks. Are there other questions or points? I had another question, about George Koob's work with the dark side, and is this sort of, these continuing deficits, are they related to the dark side? And often people talk about motivational or cognitive changes, but I'm just wondering about persisting emotional changes and the kind of persisting negative affect, and just makes that hill so hard to climb.
So I do think there is a lot of that. There is persistence of some of the dark side, although the subjects that are abstainers that had abstained for the longest period of time did have were not as depressed and were not as anxious. And I do think that that's an important piece of their recovery, that they were better. And that was true even after 3 to 5 months. Those participants had less issue than in those kinds of things. So hopefully those are things that can recover.
Mm-hmm.
It may take a long time, but I think there may be some hope.
And I had a question about one of my favorite areas of the brain, is the habenula. So my former postdoctoral advisor, Gaylord Ellison, in one of his last sets of studies showed, it's in the rat, but be that what it is, that stimulants, including cocaine, at fairly high doses, but that there is neurotoxic damage, excitotoxic damage in the habenula, and that this kind of defect in this feedback inhibition from the telencephalon back to the brainstem was something that was fairly permanent. Have you looked at the habenula at all?
So in our animal studies, we certainly look at the habenula, and there have been, I'm trying to remember exactly whether we saw residual effects in the habenula. I'm guessing, I'm thinking that yes, there were in the studies in which we looked at the residual effects, deficits that were associated with cocaine, that there were still some effects in the habenula. But habenula is definitely one of the places where we see effects, very strong effects in on decreased functional activity in the habenula as a result of cocaine. It's a little hard to be able to distinguish maybe medial from lateral habenula and the connections, and whether we're looking at the lateral habenula or whether we're looking at medial. It's a little difficult in these animals, so I'm not going to try and parse it that closely.
And when it's one voxel, it's hard to say. It's like,
yeah, I know, it's pretty small, so it's pretty hard to figure out. In even with autoradiography, it's still tricky to be able to distinguish.
Right. Well, thanks. This was just a marvelous discussion.
Great fun.
On top of a great talk, and I really appreciate your coming and speaking with us today.
No, it was fun. I really enjoyed it, and thank you for having me. I really appreciate it.
Sure.
The opportunity.
And I wish you were here, and we could take that.
Yeah, I wish you were here.
Anyway, so I just wanted to remind people in 2 weeks we're going to reconvene at noon, and it'll be Dr. Brian Jones from University of Tennessee. And have a great rest of the day.
You too. Thanks very much, everyone.